Good morning, good evening, and good afternoon, everybody. And thank you for that wonderful introduction. So my talk is a study on tick-borne diseases in neotropical animals in Trinidad and Tobago. And as you said, I'm from the University of the West Indies, which is located in Trinidad and Tobago. So this study has been published in Parasite and Vectors. And I just want to acknowledge my collaborators and co-authors from the Koret School of Veterinary Medicine, Hebrew University of Jerusalem, Rehovot, Israel, Professor Harris, Dr. Gutierrez, Ms. Yaret Nachambaila, and Professor Gad Barnett. So my talk will just take you through briefly an introduction into the study, and of course I'll go through my methods, and then I will give you a little bit more detail in my results and discussion. So what are neotropical animals? So this study basically looks at the mammalian wildlife in the neotropics or nootropics, which is basically the South American type continent. And these animals have not really been studied much, and little is known about the diseases that they harbor and the diseases that they can transmit. So this study aims to just shed some light on looking at the hemoparasites that are currently circulating in the wildlife population, especially those in Trinidad and Tobago, which have not been studied at all. So which neotropical animals are we referring to? And you will see later on why we picked or we chose or we studied these particular species that are commonly known as the agouti. There's the wild hog, there's the opossum, the red-brocked deer, and the armadillo. So these species are commonly hunted in Trinidad. So they're actually harvested from the forest for food for human consumption. As you know, wildlife have been known to harbor quite a lot of diseases. And these diseases, especially those transmitted by arthropod-borne vectors such as ticks, can of course affect domestic animals and human beings. And Trinidad is no different from most of the other countries where you have a lot of deforestation and encroachment on these habitats, therefore increasing the possibility of exposure to the arthropod vectors and the diseases that they transmit to both the domestic animal population and the human population. So where is Trinidad and Tobago located? So Trinidad and Tobago is located just on top of Venezuela. As you can see, it is close to that South American mainland. And that's very important because Trinidad's flora and fauna are extremely similar to that of the South American continent. Unlike our sister island and the other islands of the Caribbean chain, which tend to have a volcanic nature and a very simple flora and fauna. So the problems that are encountered in Trinidad especially are not often seen when you speak about neotropical animal diseases, are not often seen or even known to exist in the other Caribbean islands. So this is extremely important because of this diverse flora and fauna. There is quite a lot of hunting activity taking place in Trinidad and Tobago. And as a result, the hunting season has been basically modified in that it's not a year-round occurrence. In order to conserve our flora and fauna, the hunting season is between the 1st of October, which is like almost tomorrow for me, and the end of February. So it's a narrow season. And therefore, people who, it's part of the culture of Trinidad and Tobago with a lot of male bonding taking place and hunters and families. So you'll find lots of young men and their fathers or uncles use this as a very strong social activity. So they would overnight in the forest in camps in close proximity to their dogs. And this overnighting in forest, of course, increases the probability of infections being transmitted between wildlife and humans. And the type of hunting is that they sit actually or perch, people perch in the forest overnight waiting for the prey or whatever to pass. So therefore, as you sit there, of course, you're game for arthropod-borne vector transmitted diseases. Added to this, when the wildlife are caught, they are placed in crocus bags. Crocuses means like maybe like a cloth bag or cloth sack, and they're carried on the backs of the hunters or their kids throughout to the forest back to the camp. So therefore, that close interaction between the carcass, especially if it is harboring ticks or any other arthropod vector, they can easily affect the person transporting them. So therefore, as I said before, there has never been any study looking at the arthropod-borne transmitted hemopathogens or any other pathogens in these types of animals. So therefore, we wanted to determine which ones are present in the animals harvested from the forest for human consumption. And of course, we wanted to therefore screen the wildlife that are normally captured for human consumption. And we chose these diseases, Anaplasma, Babesia, Bartonella, Ehrlichia, Rickettsia, and Theileria. And we chose to try to find them or to screen for them using molecular methods. So we know from previous studies, especially studies done in South America, that certain neotropical animals will harbor some of these pathogens. So therefore, we surmise that if it's present in the South American fauna and flora, they may also be present here. And it's also important to note that these pathogens are transmitted by various ticks and other arthropod-borne vectors. And also, many of these pathogens are zoonotic, especially Anaplasmas, Ehrlichias, and the Bartonella, and the Rickettsia, and some of the Theilerias. So samples were collected over 2 consecutive annual hunting periods, that is between 2017, 2018, and 2018, 2019. Hunters were contacted and asked to collect EDTA blood samples on slaughter. So animals are slaughtered by severing of the jugular vein, and at that time we asked them to collect the EDTA or blood samples. Also, any wildlife carcasses that was submitted at the School of Veterinary Medicine for necropsy were also sampled so that we could try to improve our sample size and collection over the period. So what did we do with the blood? First, there was a microscopic examination of blood smears. So the blood smears were stained with Reinschemsa and examined under light microscopy for the presence of hemopathogens. And then DNA was extracted using conventional methods, using the Keogen blood tissue kit. And then we performed various PCR reactions on the extracted DNA. And then samples were sent for sequencing to determine what, if it was amplicons that were generated, was sent for sequencing to see if we could elucidate what they were. So this is a list of the various primers that were used and the genes that we attempted to amplify for Babesia theileria, Ehrlichia anaplasma, Bartonella, and Rickettsia. So it was a combination of conventional PCR for certain organisms and real-time PCR. As you can see, real-time PCR was used to for one of the Ehrlichia anaplasmas, namely Echinus, and also for the GLTA gene for Rickettsia. I just want to note that if real-time PCR of amplicon was generated for the Ehrlichia anaplasma, it was subjected to the conventional PCR to produce a band for sequencing. So where were samples collected from? So this map shows that samples, as you can see, were collected really well spaced out throughout different parts of Trinidad and Tobago, from Trinidad, sorry, not Tobago, from the north to the south, to the west, to the eastern and western coast. So we were able to to collect 43 samples. The majority of our samples we were collected were from the agouti, and the rest were from the opossum. So smaller quantities were collected from the collared peccary, just one, and capybara. So, and the deer. So basically the deer and the capybara were collected at necropsy. Our results. So our results show that we were only able to amplify Babesia theileria DNA from 2 adult female agoutis. We were able to amplify Anaplasma ehrlichia DNA from the spleen of the deer. As I told you, the deer was submitted via necropsy. And of course, we were also able to amplify Anaplasma DNA from the collared peccary. We were not able to amplify any Rickettsia, Hepatozoon, or Bartonella species. So this is pretty important because these agents are actually highly zoonotic and are known to exist in the neotropical fauna of other South American neotropical animals. It's important to note that we did get some very interesting incidental findings. On examinations of the blood smear, we found rod-shaped bacteria in the neutrophils of the deer. I remember that deer was submitted for necropsy, and I'll give you a little more about that. And we also found a lot of rod-shaped bacteria and cocci in 10 smears. Okay. When we sequenced the amplicons that we got from the deer, it was about 99.8% homologous to Anaplasma bovis. The amplicon from the collared peccary appeared to be similar to Ehrlichia canis. We got very low identity, 90.4% for the amplicons from the agouti. They seem to align with more theileria species, but we couldn't really figure out exactly what they were. And the DNA from the Babesia species were amplified using all the primers. So those 2, we got it using all the different primers that we had. And when we looked at the amplicon from the red brocket, there, we were only able to, we got both spleen and blood. So we were only able to amplify it from the spleen of the red brocket there. So the collared peccary, all of them were amplified using the 16S rRNA genes that we subjected the extracted DNA to. So other studies, other South American studies show that Anaplasma species are found in the deer, and the deer is an important reservoir for Anaplasma species. So our findings were in agreement with other studies conducted in South America concerning the presence of Anaplasma species in the red brocket deer. The deer that we found, which I'll go into a little further, was sick. It was brought in by a necropsy, and we did get some of the history for it. And as I said earlier, the Anaplasma species was only amplified from the spleen and not the blood of this animal. So a little bit more on this particular deer. The deer was recovered as a chance encounter, and it was actually rescued by villagers while being chased by hunters. So it was rescued by villagers on the side of the road while being chased by hunters. And that deer was in poor condition, and they actually brought it to a veterinarian who then had to euthanize the animal because it was in such poor condition, and it was submitted to us for necropsy. So this was the only animal also in which I were able to find ticks. So, and these ticks identified from these animals were Haemaphysalis, juxtacochi. So this deer was euthanized, and it had evidence of infection, typical of Anaplasma ehrlichii. It was anemic, large spleen, etc. Very poor doer. So hunters were actually chasing this animal, which we suspect for human consumption. And therefore, we think that hunters must be educated on recognizing normal and abnormal animals and the potential risks associated with wildlife and contact with ectoparasites, because this deer was covered with ectoparasites. We noted the 10 blood smears contained occasional cocci and extracellular bacteria or rods, which may be attributed to contamination during the slaughter process. So these were the findings of the blood smears of mainly the agoutis that we found. And this is an incidental finding, and we know this is of public health significance because there was contamination, and therefore we believe that slaughter practices out in the forest may not be very hygienic, leading to hematological spread of bacteria contaminants. As I said earlier, we found that the bacteria, some of them were actually engulfed in the neutrophils of the animals as well. So therefore, hunters should be trained into safe slaughter procedures, including cleaning of knives and during butchering to avoid cross-contamination. So in conclusion, on sequencing and alignment, we suspect Anaplasma bovis in the deer. We suspect E. canis in the collared peccary and Thylaria in the Thylaria species in the agouti. We still could not fully speciate the Anaplasma ehrlichii nor the Thylaria species. However, this study is a starting point to encourage further investigation into the health of neotropical animals in the rainforest, which we know has a very close association between Trinidad society and culture. So the ticks collected were Haemaphysalis juxtacochi. And this concludes my presentation for today. Thank you very much.
Yeah. Thank you, Karla. Thank you, Karla. Mustafa, I think you are mute.
Thank you, Karla, for the great presentation. And it's time for a discussion and some few questions. I'll start with one, and that is, as you know, sometimes when people have a tick on their body, how do you get a tick to release from the skin and not to leave its parts in the body?
You have to, of course, try to get it to detach. You can use a tweezer to sort of hold the mouth parts and not the head. Okay. So that's one way of doing it. So try to pull from the mouth parts and not the body or the head.
And the importance of removing the head is
because what it is, you don't want to get the mouth parts stuck into your skin, because that can cause an abscess. Yeah. So it's important yeah.
Let me see if there are other. No, go ahead. Let me see if there are other questions in the chat, or if people can raise their hand. Can somebody raise their hand and ask a question, please?
In the chat room.
Board. Yeah.
Karla, it's a question
from
Alberto Perez de Leon asking if the ticks you collected from the deer were the Haemaphysalis longicornis?
No, it was Haemaphysalis juxtacochi.
Okay, thank you, Karla.
Thank you. I have another question for you, and that is just in general, how for the audience, how do you treat tick-borne diseases in general?
Okay, so If it's the Ehrlichia anaplasma, it's very responsive to doxycycline. And for dogs, or even for dogs, we tend to use doxycycline. And for if it's a ruminant, we tend to use oxytetracycline. There's also a drug called imidocarb, which which can also be used off-label. For Thyleria babesia, we tend to use imidocarb more, imidocarb dipropionate.
Thank you. Does anybody are there any other questions on the chat, or if you would like to raise your hand?
Thank you very much, Karla, for a very interesting presentation. I just want to ask about the sample of the animals that you collected. There were a lot of agouti, but just one peccary and just one deer. Is this an indication that these animals are like overhunted and the agouti are not really being given that much attention by hunters?
It could be. You see, those animals are also more difficult to hunt, and hunters will, I think the agouti are easier to catch. So the collared peccary and the deer are more difficult to get, and they have to have special hunting dogs who actually just go after these specific animals. So, and during that time of that particular hunting season, there was a lot of rain, and it was very difficult for hunters to delve as deep into the forest as they would normally, I would say normally do when there's not that. So there was some level of environmental deterrent due to the rains and the floods that actually prevented access to the deeper areas in the forest. So it was more difficult to get the larger animals, game animals, than in a year where you didn't have those types of environmental conditions. So that also influenced the different species of animals that we were able to get samples from during those particular hunting periods. Yeah.
Thank you, Karla.
You're welcome.
I don't see any other questions unless someone else has a question.
I see Hussein Sabir has his hand up.
Yeah, thank you so much for the interesting talk. I just want to know about the two possible, you can say, information. The first one is, yeah, we can say the deer were also infested with ticks and other, like you have found the babesia and Thyleria anaplasma species as well. But what were the possible reasons that we can assume about the like you were not be able to detect the Pathogens and Bartonella species? Either like what are the possible reasons that we can say?
Okay, so especially for the Bartonella, it could have been just a technical issue. It's thought that maybe if you try to culture the blood first before you extract DNA, you have a higher success of actually getting Bartonella. But so we did not do that. We just extracted the DNA directly from the blood. So it's assumed that to get a better success rate for Bartonella, we may need to culture for Bartonella first and then try to extract. But we were pretty surprised that we did not find Bartonella in any of our samples. And yeah.
Okay,
so thank you for that observation. Hepatozoan, there's no real, we can't think of any reason why there was no hepatozoan. We think it has been found, but it hasn't been described as commonly as Thyleria babesia and Ehrlichia anaplasma. So yeah, so we haven't seen many studies finding hepatozoan. So maybe that's just in agreement with other studies. But again, our sample size was quite small, so as well.
Yeah.
So.
Thank you.
Hey, Karla, for the audiences who are not familiar with ticks, what should people watch after a tick bite in general?
Okay, so if you've been bitten by a tick, first of all, you should look at the site of the tick bite to look for signs of infection or inflammation, because you can get a simple abscess from a tick bite. So for tick-borne disease, it will feel similar to the common cold, where you probably get a headache, not feel too good. And then later on, depending on the disease, if it's a rickettsia, you may see a rash turning up, okay? And that is something that you have to look out for. And fever, joint pains, general malaise, that's generally the clinical symptoms in people. So if you've been bitten by a tick and you start feeling feverish, or if you're getting a rash coming up on your body, joint pains, then you should really seek medical attention immediately.
Thank you. We have another question from Dr. Beto. Would you like to ask that question, or let me ask it for you? Go ahead, Dr. Beto.
Thank you for the very interesting presentation, Dr. Georges. Quick question: what's a follow-up, or what do you plan to do next with your research program on tick and tick-borne diseases in Trinidad and Tobago, based on your findings? Thank you.
Okay, so for these findings, we really need to, well, we have a lot of ongoing programs going on right now. So we do have a project where we are looking at the ticks itself and subjecting them to sort of like an array to look for various pathogens within those ticks, from viruses to bacteria to other protozoan. So we are doing that. It's called a biomark system. And we also have someone looking at deep sequencing of the ticks, looking for viruses, different types of viruses. So those are the main things. Looking for the pathogens in ticks, those are the main things that we are looking at, and we also want to translate that research. That research is more on ticks on domestic animals and companion animals. So we would really like to translate that to ticks found more on wildlife, to determine what exactly are there in these species.